just want to say thanks before I get started to the entire SP staff. I think this forum is a great way to share and learn. So appreciate the chance to talk to everyone today. We think we have something unique in the lyosphere and lyobead space in terms of how we make these beads. And so I'm interested in getting to know a few of you. Hopefully there are questions at the end. Thank you, Zach. Thank you, Audrey, and the SP team for letting us present. So before we get heavily into the process of forming beads, I want to spend a little bit of time to talk about why people are considering lyobead, some of the things to think about when you're considering maybe moving to this format or using this format for the first time. Some of the whys are maybe obvious. You know, the initially when people think about lyophilization, you think about trying to have a thermally stable solution, right? Materials that can handle supply chain in a much more robust way, longer shelf life. That's sort of a gold standard for moving to lyophilization. But when you start to think about an individual bead as opposed to a cake, there's some pretty interesting aspects to that geometry that can really help. The first and maybe most obvious is reconstitution. Right now you have a 3D structure that can be reconstituted in your solutions at the point of use. That can increase efficiencies, that can increase time for reconstitution, and ultimately take away a lot of the variability that cake lyophilization can present. The other real improvement is the ability to now manipulate these beads or these doses of reagents in a very meaningful way. There's now the ability, if you're doing a kit manufacturing, or if you're trying to precisely dose the amount of active reagent into a vial, now you can pick and place these beads potentially with automation at high speed and really achieve some pretty unique things with regard to applications. Things to consider though, now this is sort of a different level of production. We'll talk a lot about the equipment that we provide at BioDot, the work that we do. If you're moving from cake to a bead, there's likely validation that has to happen. You need to think about what that does to time of market. And then you have to sort of just acknowledge that not all reagents can form a bead. So partner early, partner with folks like BioDot, SP, other contract developers in the space to sort of do that initial evaluation and just think about the time. So before I jump deeply into forming lyobeads, I do want to just make sure everyone sort of understands where we come to this point in time. So BioDot, if you don't know, we're sort of relatively new to the lyosphere and lyobead space, but we're not new to diagnostics, and we're definitely not new to diagnostic manufacturing. So BioDot's a 30-year-old business. We specialize in high precision, high accuracy, low volume liquid dispensing, and we've done that for many years. Where typically we're patterning cells or proteins or DNA onto some sort of surface. Usually for manufacturing, we spend a lot of time in next generation laboratory science where we're miniaturizing assays. But our volumes that we work in are either in the microliter range, the nanoliter range, and now even the picoliter range. So incredibly small drops, usually high rates of dispense. And all that is really focused, like I said, primarily on the diagnostic space, but we do work a bit in pharma. So all that to say, so how do we get into the lyobead? So this ability to dispense a very small, very accurate, very fast droplet brought us into this space where partners of ours realized that we had this capability, and they were sort of struggling with some homemade automation that they had built for doing lyobead. And so they asked us to participate. And so that's where the pneumatic biojet was born. And so now with what we call the biojet technology, we can dispense a single droplet at high speed but with, and you'll see later in the presentation, with incredibly spherical capabilities and consistency with regard to geometry. That's critical, right? When we're making beads, not only do you want to have the right volume, but you need to have spherical quality and consistency from bead to bead. If you have ellipsoids or doublets or things that are unpredictable, you can have issues either with dosing, you can have issues with reconstitution, and you can definitely have issues with your downstream automation when you're trying to pick and place these beads into a device. So that with the BioJet, as you can see, what's unique, and we'll talk about it quite a bit today, is that we have this ability to do what we call on-the-fly dispensing. So as we're sitting over a liquid nitrogen bath, we can deliver droplets as we scan back and forth. With a single tip, we can do anywhere from 7,000 to 10,000 beads per hour. And with 4 tips, we can achieve 28 up to 40, and sometimes up to 90,000 beads per hour. So, you know, I think in this space, that's typically been a limitation, is the throughput. You know, most automation and most homebrew systems can't achieve that level of capacity with that dynamic range, or with the dynamic range that we can achieve. And as you see here, we're dispensing typically anywhere from 2 to 30 microliters as a single drop with very, very good CV. So how do you make a bead? This is a representation of how bead formation works on our platform. You see here, this blue outer region or device is really a liquid nitrogen bath. You fill that bath with liquid nitrogen. Within that bath, you have a tray, a collection tray, and then it's important to have a partition. So as you dispense single beads into the liquid nitrogen, they have to be kept away from each other while they're allowed to freeze and then drop. Once they freeze, they will drop to the bottom where they fall into the collection tray. But that partition's important. And as you sort of see in this cartoon, what we do is we sort of scan back and forth. So we're scanning back and forth across the top of this bath assembly, moving back and forth, dispensing single beads into each partition, and then we don't revisit that same partition or that same well until we've achieved the freeze time and that the bead is dropped. Once you have your beads, it's time to remove the liquid nitrogen from the bath. So that's either done through a drain or you just let the liquid nitrogen evaporate. The assembly for the partition needs to be removed. And then you can take out your tray, from the liquid nitrogen and move it to lyophilization. There's typically a trace amount of liquid nitrogen still at the bottom of the tray. That's important, mostly because of timing. You want to make sure that once you've frozen the beads, they need to stay frozen throughout the process until they move into lyophilization. And so that you typically want to have some trace amount of liquid nitrogen just to make sure that the beads stay frozen through that process. Ideally, you're gonna do an immediate transfer where you transfer the tray directly to the lyophilizer, but sometimes it's not practical. This is what our assembly looks like. At BioDot, we typically either design our automation to an existing bath and bath assembly, or we provide this capability to our customers. So as you see down here at the bottom, we have our outer bath. We have a lifting device that just allows us to remove the materials safely from the bath assembly. We have the collection tray here, the blue device, and then the partition sits on top. And then, like I said, with the Sphera platform that we have, and we'll talk about our different automation options. It typically takes, for a single tip, it will take about 90 seconds for us to move from the beginning of the partition, well 1, to the end of the partition, well 240. And so from an application standpoint, you know, what we spend a lot of time thinking about and working with our customers on is really understanding what that freeze time is, right? So if the freeze time is 90 seconds, then that's perfect, because when we dispense the first drop, it takes about 90 seconds for us to make our way through the partition and then dispense the last bead into the last partition hole, and then we can revisit well one, because we've had sufficient time for freezing. If the time is longer, if it takes longer than 90 seconds, then we have to institute a pause, which ultimately slows down the process. If I add more tips, I can move through the partition faster, right? If I have 4 tips dispensing simultaneously into each of the partitions, I can move faster, but now the gating step is really the freeze time. So we need to sort of balance that. We spend a lot of time talking to our customers about the size of the bead, the freeze time. Typically, as the bead reduces in size, the freeze time reduces. And so for our customers that are working in the sort of 2 to 4 microliter range, the freeze times are in the 30-second range. As you grow in size, you tend to grow that freeze time, and therefore that has to be considered. I want to show some beads. So this is a top-down view of that partition. So you can see that this is post-process, and so we've already dispensed beads. We're typically doing anywhere from 10 to 20,000 beads, depends on the size, into a tray per run. The beads don't have to make a monolayer. You can usually do 1 or 2 layers of beads in the collection tray, so you can collect quite a few beads. But again, this is what the beads may look like as you look down through the partition. Hopefully people are starting to understand the importance of the partition. You'll see it. We have a video later that we can review as well. Once the partitions are moved, now you see the beads themselves, frozen and in the liquid nitrogen. This is the tray that would go directly into lyophilization, and these beads would be lyophilized together. Do you have some videos? So this is an example of our platform. The Sphera, we'll look a little deeper into it on a few slides, but you're going to see a single channel dispensing over the beads, and then the camera is going to move over the top of the partition, so you can see what the beads look like as they're floating in an individual partition. I believe the droplet volume here is 10 microliters. If you look closely, you're already seeing some of the beads freezing and floating in that first row. And then once the freeze time is achieved, they will freeze and then drop. So we don't see that yet in this video, but that's what happens during the process. So kind of fun. Fun to see these beads in real life. It's an interesting process to watch. And then this is just another example. Sorry, that was loud. Of a different instrument and a sort of different process, but you can see same concept. We need to dispense into the individual partitions and to make sure that the beads don't collide with one another until they drop. We want to make sure we're not creating any doublets or triplets during this process. When we're forming these beads, and we have some imagery that you'll see later where we actually can see these beads or the droplets in flight. The technology that we use, we call it the BioJet. We basically have integrated a pneumatic back pressure system with what we call our BioJet valve, which is basically an inkjet valve, the same inkjet valve that we use to dispense nanoliter droplets. We combine that with special tip geometry. So you see we have 3 options here. 508s, the 762s, and the 1000 micron orifice sizes. So we will every chemistry is a little bit different. The rheology and the viscosity of the reagent matters, whether you're cooling the reagent or dispensing at room temperature definitely matters. And so we'll select the proper tip geometry for the application and the size. And so we have stainless steel versions of our tips. We have different materials that we'll use, different surface coatings to enable the spherical droplet that we'll take a look at later. An important piece of our technology is the horizontal drop camera. This is a very useful tool. We use it for drop optimization for settings. Basically what happens is the machine will move into this region of the instrument. So this is, you don't have to dispense a bead to understand the consistency of the droplets that are coming out of the instrument. So we have a horizontal drop camera. It's looking sideways, and as you see here on the right, we will park just above the camera and we'll start to dispense. And so we can actually capture, we have a stroboscope or a strobe light that is opposite of the camera. And when we send a trigger signal to the dispenser, at the same time we send a trigger to the strobe. And so what it allows us to do is capture the droplet in flight. I'll show you some images in a second. But this allows us to characterize settings, right? We're tuning the dispensers to make sure that we have a proper volume and a proper dispense. And this is just a very convenient way to do process optimization and potentially in process QC. So this is the image. This is the image of a drop. As you see here, a 10 microliter drop in our software. These are live images that can be recorded. It's hard to see in this view, but we also capture statistics. We can actually calculate the volume of that drop. We assume symmetry around the z-axis, and so we integrate that volume or the size of the drop to calculate a volume. And then ultimately we can look at CV, so precision of drop to drop. It really is the best way to tune CV settings and also velocity. We actually know, because of the timing of the stroboscope, we can actually adjust that, so we can actually see the drop at different levels, right? And so we know the height in which we dispense above the surface of the liquid nitrogen, and so we can adjust the settings in the drop camera so that we can see, okay, this is what the drop is going to look like exactly when it hits the surface. In some cases, when it's just coming out of the nozzle, it's not perfectly spherical, and so you need to give it some time to sort of form that drop in flight. But again, we can change that offset or or the delay with respect to the camera, and that allows us to see what that droplet actually looks like. And so you see this is a very nice spherical drop. This is an 18 microliter droplet in flights. We do have a video. Luckily, not a lot to see because we have you can see it moving a bit, but you see that there's incredibly consistent shape, right, of that droplet from drop to drop. And if you look to the right in this section, you're actually seeing that we're capturing the data in real time. So the droplet volume is 18.1 microliters. We calculate standard deviation, and then we have a CV. So the CV is less than 1%, approximating 0.3%, which is quite good. Here's some statistics at 4.2 microliters, 1,000 drops, similar to the video. We are our calculated CV. You can see it's a very tight distribution across those 1,000 drops. We're very excited to have that sort of capability that is deployed in this application, and we have a CV of 0.52% with very little drift. Talk a little bit about the options of platforms that we provide. We have an instrument that we're excited to be launching we launched late last year called Sphera. This is a benchtop platform, really used for sort of moderate-sized production and R&D and process development. You can have 1 to 4 channels. It has the horizontal drop camera, as you see on the right. It has an enclosure to, again, for people that are new to this, we are working with live liquid nitrogen, so incredibly cold temperatures, right? Safety is key, so we have an enclosure to protect from splash. And then we've designed, we can either work with your tray design if you have one already in place, as long as it fits on the platform, or we have a standard, as we showed, standard bath assembly, and it happens to have been designed with the team at SP, and so the size of the tray, the collection tray that collects the beads, fits directly into one of the shelves of the SP Advantage Pro. Then we have a larger system that we use for high throughput production. So again, going back to that concept of freeze time, if your freeze time is 90 seconds and you can get through a partition in, let's say, 45 seconds, you actually need more partitions, right? You need more holes, or you want to have more holes that you can shoot into, so that cycle time can match the freeze time, right? The dispensing cycle time needs to match the freeze time. And so that's how we achieve, this is how we achieve that in high production. So you can see in this case, we can hold up to 2 large baths. These baths are actually larger than the SP formatted or Advantage Pro formatted system. Typically, we're always using 4 channels, and we're dispensing, again, high throughput, but the concept is the same. This is a shuttle out here in the front where the baths can come out, so an operator can access the liquid nitrogen bath and the trays, and then when they're ready to load, they come back in and they can dispense. So, throughput on this system is usually in the 60 to 90,000 beads per hour. This is just a top-down view of the dispense head. So, of the machine. So we have the dispense head in the top. These are the 2 baths with their partitions, drop camera. So very similar design to the Sphera product, and that's really the intent. We always build our R&D platforms so that you can directly apply those learnings and the process development to a high throughput platform. So the technology is the same, the way that we monitor the beads is the same, and basically all we're doing is expanding that capacity with respect to the bath. Like I said, everything's really cold. So we do spend a lot of time thinking about temperature and not freezing our beautiful automation with liquid nitrogen. So the bead, the tray, and the bath assembly, they make intimate contact, obviously, with the instrument, and so we have installation strategies that have been put in place both on the Sphera platform and the larger 86,000 platform that we use for production. So we want to make sure that everything's properly thermally isolated, and that's important. Safety is a major consideration, and you want to minimize any transfer of temperature to the automation. And then, we're actually quite fortunate. In 2001, we were acquired by ATS. We're now part of the ATS family of companies. SP was more recently acquired by ATS. Interestingly enough, all 3 organizations were independently working very hard on the Lyobead question in each of our lanes, right? We were forming beads. SP is an excellent lyophilization technology that has been in place for many years, and ATS has incredible automation for doing high-speed pick and place of the beads once they're frozen and putting them into devices, right? Either for pharmaceutical applications or typically for diagnostics. And so we're very pleased. We work very closely with our partners at SP and ATS to build end-to-end solutions for customers, so we can go directly from an instrument from BioDot to lyophilization to the final product, all with one supplier. So, and the applications typically on the bead placement side are often custom, right? The devices are never the same. The applications are usually unique. So BioDot and SP can typically provide an off-the-shelf solution where ATS will work with you very closely to design the application and the throughput to meet your needs. And I think I raced through this. I do want to have a lot of questions. Hopefully it's a sort of a new technology and sort of a novel way of forming beads. So if you have any questions, you're welcome to contact me directly. If you have general inquiries about BioDot or bead formation, you're welcome to go to our website. Yeah, like I said, I'm happy to answer any questions if there are some. Hopefully you enjoyed the time. I think we're, like I said, we're quite pleased to be in the space. The lyobead formation and lyosphere formation is a growing trend in the industry, and so we want to learn as well. So thank you for your time, and we can open up to questions.
Okay, thanks, Brian. It looks like we do have a couple of questions already coming in. Again, I just want to bring everyone's attention to the questions box, which is right in that main channel as you're tuned into the webinar. So feel free to submit a question and we'll try to get to them all.
Okay, sorry, this is a new format for me, so let me fix my view. So what are the challenges for sterile manufacturing of lyobeads? Good question. So sterility is definitely a requirement, specifically usually for pharma. I think it's important to note that our systems can and will fit inside isolators or fume hoods. For us, the challenge or the thing to consider is how do you sterilize the fluid paths, right? These reagents need to be sterilized with the fluid path, right? The path that the reagent takes from the bulk container. There's a bulk container that the material goes through our dispense channel and then dispenses out the end. Those, if you're working in a sterile environment, those dispense channels can either be independently chemically sterilized, or you can segregate. You usually do both. You can segregate a certain fluid path for a very specific chemistry. Do we need the liquid nitrogen to be sterile filtered? How do we achieve that? In our applications, or the applications to date, we don't require that. Most of the work to date at BioDot has been in kit formation or chemistry bead formation that doesn't require that level of sterility in the liquid nitrogen. I don't know off the top of my head if that's actually a need based on the temperature. So we've been typically working with a standard liquid nitrogen source. So we have Airgas bring in liquid nitrogen. There's sort of two ways to load the bath. The bath on our design has an inlet filler, so you connect to your supply, and then there's a valve that allows you to basically fill the bath through the valve control, and then you stop that when you're ready to run. Or that people have used dewars, so they'll first transfer first transfer the liquid nitrogen into a dewar, and then from the dewar into the instrument bath. Is the material construction of the partition of critical importance? It is. First of all, it needs to withstand the the rigors of liquid nitrogen. So we're typically working with aluminum. You want them to be precise. You want them not to change over time, because if you think about the process, we're scanning over these partitions at high speed, and if there was great variability with respect to the partition, then we can't rely on the position of that. We can't rely on the position of that partition or the size of that partition, and so that could affect throughput. You may have to do what we call step and repeat as opposed to on the fly, so you could have issues. So high quality, either stainless steel or aluminum. There's a question again about sterility, and I think we sort of touched on that before. So again, system can depend on the size. The Sphera unit for sure will fit inside an isolator or a fume hood, and then we would chemically sterilize the dispense channels, and we would typically dedicate a fluid path from beginning to end to a particular product. Let's see. Sorry, I'm scanning through. Is the dispensing system moisture, humidity controlled, or can it be? Good question. So the standard platform, the standard Sphera isn't. The enclosure is really just to control splash or to protect from splash. But we do many humidity controlled environments at BioDot for a lot of our platforms, and so that could certainly be added as a custom feature for sure. Let's see. There's a question about FDA requiring liquid nitrogen to be sterile for pharma and biotech applications. Good segue into sort of the question about developing applications and platforms. So we at BioDot do not do the application development. We're experts at doing the liquid dispensing, and so we typically work with some other contractor or our partners to manage the needs of either regulatory bodies or the application. So I don't know what the FDA requirement is with regard to sterile liquid nitrogen. We would work with our partners, and then when it comes to development of processes, right? How do we lyophilize these beads? What are the right constituents in the chemistry to promote lyophilization? Those are usually done at partner sites. Let's see. So the question is that we showed the part leading to bead formation, and we mentioned partners for the rest of the workflow. The question is whether we have the entire workflow available for customers to handle lyobeads. And so At BioDot, we have a SP lyophilization system, the Advantage Pro. So you are welcome to work with us to do demos. If you know your process, if you know the conditions for lyophilization, you can certainly come to BioDot, and we can do demonstrations on our platform, and then take those beads all the way through lyophilization. Automation would happen through our partner ATS. They have sites in the US and in Canada, and then globally in Europe as well. So we would have to sort that out. We'd have to maybe think about either a 2-site demonstration or potentially ship some of our equipment to an ATS site so we can do the full demonstration. Is static electricity a challenge when handling the frozen and/or dry beads? Yes. So the beads are light. They will stick to things. So there are lots of typically in the process there are lots of areas where you'll want to deionize your materials. We don't work with a lot of plastics. Plastics tend to carry charge, and so we try to keep things in, like I said, in the stainless steel carriers as long as we can. But there are lots of deionizers. There's area deionizers that you can use to help with that reality of life. Static is not usually, we haven't seen it to be an issue when we dispense. It's really the post lyophilization where static can be a challenge. There's a question about friability of the beads. Have we encountered challenges with friability of the beads after we lyophilize them? That really goes into development. And so, like I said, we have both with SP and with sort of our network of partners. I think we can get you in touch with folks that if that is an issue that you're encountering now. I don't think the method that we create the beads would have much of an effect. That's probably more with formulation or possibly the lyophilization process or the settings for the lyophilizer.
Okay, getting a lot of good questions. Thanks, Bryan, for the thorough answers there. It looks like that's all we have for now, but I will give it a couple more minutes to see if any other questions come in. We do have some time, so again, if you have a question, feel free to submit it in that questions box, and we'll hang out here for another minute or two.
Yeah, really appreciate the time, and like I said, I would encourage anyone who's curious to reach out, and then we have platforms here. I'm happy to do demonstrations, product demonstrations, and do feasibility work here in Irvine. I'm sorry, I should have said this before. So BioDot's headquartered in Southern California, in Irvine, sort of tucked in between Los Angeles and San Diego. And then we also have a facility with Lyobead capabilities in the UK, so in Chichester, in the south of England.
All right, great. Well, that looks like it's it for now. So in that case, thank you everyone for your attendance and participation in today's webinar. And again, a special thank you to Bryan for volunteering his time today. If you've missed any part of today's presentation or would like to invite a colleague to attend the next live session for today, we are going to have a repeat session at 4 p.m. Eastern Time. And as a reminder, a recorded version of this presentation will be available after 24 hours at our webinar channel, which is gotostage.com/channel/SP Lyo University. Thanks again, everyone, and enjoy the rest of your day.
Thank you, everyone.